Hello there, this is Rohan Mishra from Bioinformatics Center, and in today's lecture, we are going to talk about one of the alternative method that is used for tertiary structure prediction. We call this method as ab initio structure prediction method, right? So just to maintain the continuity of our previous lectures, well, let us start out with revising how and when you should use this approach. Okay, so as per the logic of structure prediction that we were following from previous few lectures, it says that uh, if you are able to find out the structure of a given protein using a technique like homology modeling, then that would be the best approach and most accurate approach to figure out the structure of the given query protein as such. Okay, but what if you are unable to find a suitable homologue for the given sequence, and yet you failed after performing secondary structure prediction, you failed to recognize a certain fold. So you won't, in that situation, you won't be able to perform this homology modeling or the fold recognition approaches as well. And in such conditions, there is only one way remaining, and that way of solving the structure would be using your ab initio protein structure prediction. Okay, so I would advise you to use this ab initio protein structure prediction algorithms, or use this methodology only if nothing works. Okay, you are advised to use this when homology modeling is out of picture and somehow you also fail to perform this fold recognition. Okay, you are unable to figure out what might be the fold for my given protein. And if you are quite desperate that you need the structure of a given protein, then you should go for this structure prediction using this ab initio method. Okay, so let us move on to our discussion on what this ab initio protein structure prediction is. Okay, so here is what we are going to deal with in your next few lectures. Okay, so I would first introduce with the concept of what is ab initio structure prediction. We would be studying about some algorithms that are usually that are popularly used for predicting the structure using ab initio approach. One of the group that is very famous in proposing the software that uses ab initio protein structure prediction is that of Simon and Baker lab, and they proposed these softwares back in 1997 to 99. Okay, or you can say in early 2000s. Okay, and we are going to deal with more of this on how this algorithm works. Okay, so let us begin our discussion. So as it is now a common understanding between us that we know that protein folds into unique structures. Okay, unless and until the protein acquires a unique structure, it cannot function. Okay, the functionality of protein is completely dependent on how do they fold. Okay, so all in all, what I want to say is that this protein functions when they acquire a unique structure. Okay, so in PDB, there are there is tremendous diversity of how many or you can say there is tremendous diversity of different protein folds that you can observe, that are known. right? And over here, there are only there are only three representative folds. All I want to show in this case is that we can say from these three structures that they have a lot of diversity so far your protein folding is concerned. Okay, so uh, next concept that you should understand in order to deal with the ab initio protein structure prediction algorithm is that you should now appreciate that the native structure of a protein are most likely to be present when they are at their lowest energy state. Okay, so what this graph represent over here is that what if you try to plot energy change on the x-axis and time on the x-axis and energy change on the y-axis. Okay, so well, this is what this energy plot would mean. Well, say for example, say you begin with any protein structure with, say, it is folded this way and it is associated with a certain energy value that is present over here. Okay, and then after some time it might change its conformation and you might end up with say some different conformation over here which is associated with this much of energy now. Okay, similarly the protein can again acquire some different conformation and gain some structure which might look something like this, and it might have some energy associated change which would end up over here. So this plot, what you say, is called as the energy landscape. Okay, so what I mean to say from this discussion is that in any given time there are many different possible conformation that a protein can acquire. Okay, and each of the conformation can be associated with change in free energy. Okay, therefore these multiple energy states and conformation can be represented in such energy landscape. Okay, so now in this energy landscape you can figure out that at some points the energy is low and at some points the energy is high. Okay, so uh, the points at which the energy is low, well, is called as you can say we can say that these are local energy minima. Okay, and in your this energy landscape there might be some point where the free energy associated with that structure is the lowest, and we call such energy state as global energy minima. Okay, so the conformational state at which there is lowest free energy that is associated with the structure of your protein is called as global energy minima. Okay, so the concept is that whenever the protein acquires the functional state, well, it is said that it is usually associated with the lowest free energy value. That is, when the protein attains the global energy minima, then it is said that it might be functionally active. Okay, so in other words, you can say that proteins acquire the functionality when they attain the global minimal state. Okay, so now I hope you understand what is a local energy minima and what is global energy minima. So the global energy minima corresponds to the lowest free energy state of the given protein structure. We can call it as global energy minima. Okay, now you are comfortable with the concept of this local energy minima and global energy minima. Next thing that you need to know is that let us now talk about what is ab initio protein structure prediction. Okay, so these were the underlying concept that we are going to need when we talk about this ab initio protein structure prediction. So what exactly is this ab initio protein structure prediction? This ab initio protein structure prediction answers a classic protein folding problem. Okay, so what exactly is the protein folding problem? The protein folding problem can be stated as: if you have been provided with the amino acid sequence of a given protein, will it be possible for you to find its structure? Will it be possible for you to figure out how that protein would fold? So this is called as the protein folding problem. Given the amino acid sequence of a protein, try to figure out how the structure of that protein might look like. Okay, so now why one would like to do such a thing? Okay, why would one like to figure out or predict the structure of any given protein if he has been provided with the sequence alone? Okay, so for example, we have lot of known structures now in PDB. Okay, there are structural genomics initiatives that provides a tons of structures. Then why would one want to predict the structure using this ab initio approach? Okay, so you could answer such questions saying that we would do such ab initio protein structure prediction. Why? Because there might be a lot of structures, a lot of new sequences whose structures are not available. And using these in silico techniques like ab initio structure prediction, you can build up the structure of such sequences, right? So ab initio protein structure prediction tells us about the concept of protein folding, like how proteins go from sequences to structures, right? So it would help us understand what are the rules that protein follows when they fold. Okay, it gives us an idea about the very physical chemistry of the protein folding process. Okay, so it imparts us with the biophysical knowledge to understand how protein fold and eventually how protein functions or how protein works. So that is very important aspect of understanding the protein physiology, right? So that is how it is very necessary that we sometime need to predict the structure using ab initio approach. Okay, so now in order to understand this ab initio protein structure prediction concepts, you should be aware of the two main concepts that are involved in this protein folding. Okay, so the very first concept that is very important is the Levinthal's paradox, and another concept that is important is called as the Anfinsen's experiment. Well, now let us first try to discuss it for a while, about what exactly is Levinthal paradox and how it is usually associated with protein folding. The same we would discuss with the Anfinsen's experiment. Okay, so uh, let us talk about this Levinthal paradox first. So Levinthal proposed this paradox back in 1969. So he was the one who threw out a question as to how does protein know how to fold. That was his basic question that he was interested in answering. he was interested in finding out ki how is that protein knows what its structure should be. Okay, so this is what his approximations were. Okay, what he approximated was that on an average a cytosolic human protein or any protein which is present in cytosol is usually 300 amino acid residue long. It would contain say some 300 amino acid residue. Okay, so we'll say for example if this is the backbone of your protein, okay, and we would say that this is the side chain. Okay, right, this is the side chain. Okay, so now if this is a normal 300 amino acid residue protein, then now you know that the backbone of these amino acid residue can acquire secondary structures like it can be either in helical form, it can form either strand or it can form loop, right? So these are called as secondary structures. So the backbone of your protein can acquire one of the three secondary structural state. Okay, so now each and every residue that is present in your protein can adopt one of the three secondary structural state that you know, right? So now if we go by simple mathematics and try to calculate as to how many conformations do the protein might have to try in order to acquire the functional state. And the mathematics is quite simple. We have got three structural state, and how many amino acid residues are there? There are 300 amino acid. So in order to try out each and every possible secondary structural state, the protein has to try out 3 raised to 300 possible combinations. Okay, and if you try to simplify this 3 raised to 300, you would end up with number like 10 raised to 143. So there are, you know, if the protein is 300 amino acid residue long, and if it wants to try out all the possible three structural states for each and every amino acid residue, it would have to try out 10 raised to power 143 combinations, right? And this 10 raised to power 143 is a ridiculously large number. It is a very big number. Okay, now if we consider that the protein tries these 10 raised to power 143 states, still we are only focusing on the backbone of your protein molecule. Okay, but it is the proteins are not only made up of the backbone, it also contains the side chain, and these side chains can also have those rotatable bond, and they can also have say some flexible conformations that the protein might want to try, right? So if this number, this huge number 10 raised to power 143 is what we get when we consider only the possible combinations on the atoms that are present in the backbone. We are still ignoring the rotations that are involved in the side chain of the protein molecule. So this number would again increase if we consider all the possible rotations of side chains as well, right? So the uh, this is what the approximations are, right? So if you want to consider all the three states, helical state, strands and loops, along with the side chain rotations, the number would eventually be far more greater than 10 raised to power 143. But the problem is that the total number of the particles that are estimated to be present in universe are in range of 10 raised to power 80. right? So if we go by the same mathematics, it would take the age of universe to try out each conformation, right? But what we know is that the proteins fold in just even a matter of seconds, right? Then the paradox now arises is that how the protein find its folded state when there is so large conformational space that is available for that protein to explore. There is such a huge combination of lot of structural states that the protein can take up. And how is that this protein find out what is my folded state? Okay, that is the question. Okay, so what do we mean by large conformational space? This large conformational space would indicate the possible number of combinations that the the protein can acquire. Okay, so that conformational space is very big. Okay, and this is what the paradox state, how the protein finds its folded state when it has got such a huge number of combinations to try. Okay, so the same question is somehow needed to be addressed in computer when we try to predict the structure using ab initio protein structure prediction methods. Okay, so this is what we need to simulate in our algorithm, in our protein structure prediction program, in order to find out how the protein might fold using this ab initio protein structure prediction method. Okay, so this was about your Levinthal's paradox. It says that there are a numerous number of conformation that protein might want to try in before it figures out uh, this is my global state, or this is my folded state. Okay, and this number is ridiculously very high, right? So that is what I want to discuss in this Levinthal paradox in protein folding context. Okay, so now let us talk about another concept that is needed in our ab initio protein structure prediction, that is related to your Anfinsen experiment. Some people also refer it as to Anfinsen dogma, and for proposing this he got Nobel Prize in 1970s, right? So what he did is that he tried to find out a solution or an answer to the question, as if what is it that controls the protein folding. Okay, so he was behind finding out the answers to the questions like, does the protein synthetic machinery is the factor that would help that protein to fold? Well, say for example, do these ribosomes or RNA play some role in guiding the protein on how to fold as such? Okay, or is it its environment that plays some role in protein folding, as to when the protein is synthesized on the rough endoplasmic reticulum and then it might move into some vesicle to, and that vesicle might go and fuse with Golgi complex. So the environment in these different cellular structures might be different. So is it possible that this environment that is present in this endoplasmic reticulum or Golgi apparatus where that protein ends up, well, does that environment has to do something with protein folding? Okay, or simply does the protein itself know how to fold? Okay, so that is what Anfinsen's objective was. Okay, so this is what he finally proposed after doing his famous Anfinsen's experiment. He put forth the hypothesis that all of the information that is required for the protein to fold is contained in its amino acid sequence. Okay, so it is not dependent on the synthetic machinery, or it is not dependent on the environment where the protein exists. for finding out or to figure out how it would fold. All the information that is required for the protein to fold is present in its amino acid sequence itself. And people usually refer to Anfinsen's hypothesis as thermodynamic hypothesis as well. Okay, so this is what Anfinsen did. Okay, so Anfinsen considered ribonuclease A as the protein model for his protein folding experiment. Okay, so what he did is that he kind of used this ribonuclease, which is, you know, a single polypeptide chain. It is a very small protein which contains only 124 amino acid residues, and when this protein folds, it forms four disulfide bonds as they are shown over here. 1, 2, 3, 4. It has got 8 cysteine residues, and these 8 cysteine residues can form 4 disulfide bonds that eventually enable it to properly fold and perform its function. Okay, so now what he did is that he took this ribonuclease A protein, and he heated it or added some reducing agent so that these disulfide bonds can be broken, okay, by addition of urea. And after heating and after addition of this reducing agent, he observed that this ribonuclease A no longer attains its or that ribonuclease is no longer able to perform its catalytic function. Okay, so it is unable to hydrolyze the single stranded RNA molecule. Okay, so we say that when you heat or add urea and reducing agent, you obtain something called as the denatured form of the ribonuclease A, or you can also call it as you can obtain the unfolded state of the ribonuclease A protein. Okay, then what he did next is that he cooled down the same protein. He removed the urea and the reducing agent, and what he observed is that after cooling down and removal of the reducing agent and urea and everything, the protein somehow acquired its functional state. Okay, so after removing the reducing agent or the urea, he observed that there is reformation of these disulfide bonds, and the protein gain its native state and it again become functionally active. Okay, and all of this experiment were performed in test tube. Okay, this experiment was were conducted out of cells. Okay, so what do these experiment indicate? This experiment indicate that the cellular machinery do not matter when the protein folding is concerned. Okay, so protein folding is independent on what is present around your protein molecule. Okay, he kind of demonstrated that the protein folding is purely a physical process. It is purely a physical phenomenon, and the protein itself know how to fold and function. Okay, if the protein folding was dependent on, say, some cellular machinery, then it would not have regained the functional state when we perform this experiment in the test tube, where there were no cellular machineries present around that protein molecule. Okay, so that is what we want to confer from this Anfinsen experiment. Right. So this Anfinsen experiment enabled us to understand how the protein folding happens, and it is the cellular machinery that is usually present around the protein is not the one that affects the folding of your protein. Okay, that was the take home message from this Anfinsen's experiment. Okay, so now one would like to have some discussion like, are there any exceptions to this thermodynamic hypothesis or this Anfinsen's hypothesis? Okay, yes, there are. Okay, so we would just discuss the three of them. Okay, it is observed that why do some protein needs chaperone for folding? If the protein knows how to fold, if all the information is contained in its own sequence as to how to fold it, then why do some protein needs chaperone proteins for their proper folding? So we'll say, for example, one of the very famous chaperone protein is in form of this GRO ELES complexes. Okay, so these GRO ELES complexes are very large container that are present in cell, and this container have a kind of lid, and this lid would contain some hydrophobic surface that are present inside this protein. There is a hollow space that is present in this GRO ELES complex, and this hydrophobic space is lined up by uh, this inner surface is lined up by the hydrophobic amino acid residues. Okay, and once the unfolded protein which needs some chaperone activity or needs some help in finding its folded state, it enters into this GRO ELES complex, and this GRO ELES complex would help the protein to refold if it is stuck in local minima. Okay, so well, say for example, if you go back to your energy landscape, sometimes it might happen that this protein might stuck up in, say, some local energy minima, and it might become difficult for this protein to cross this energy barrier and attain this local minima as such. Okay, so in such a case, there are chaperone proteins that would help this particular protein which is stuck in this local free energy minima to refold around itself and obtain the global free energy minima. Okay, so this is what the function of this GRO ELES complex is. Okay, the second example might be like, there are certain synonymous codons that are known to direct the co-translational folding. Okay, so it has been observed that there are certain codons, and if certain codons appear while the process of translation is happening, it would appear that these codons affect the rate of protein folding. Okay, certain codons slows down the process of protein folding. Okay, the third case that I would like to discuss is about this thrombin. Okay, so you know that this thrombin is usually generated in its precursor form, right? And this precursor form of thrombin needs to be cleaved. Okay, and when certain peptide bonds from this precursor of thrombins are cleaved or cut off, it would gain the functional stability, or it would gain the functional form. So that is the case with many viral genome polyproteins as well. So these viral genome polyproteins are synthesized as a big protein, and then they are cleaved into smaller proteins which gain the functional form later on. Okay, so now it is said that cleavage of, say, some peptide bonds might bring that protein to its lowest energy state, and when the protein acquires the lowest energy state, it would function properly. Right, so these are exceptions to your Anfinsen's hypothesis. So the bottom line, or the take-home message that I want to convey you, is that the thermodynamic hypothesis that was proposed by Anfinsen holds true for almost most of the globular proteins that exist in cell. Okay, it is not that each and every protein would follow it, right? Another take home message would be that these proteins would need to get into its lowest energy form so that it can function properly. That is what I want to discuss. Okay, so now let us talk about this ab initio method as such in protein folding concept. So what have we learned so far? Okay, so far the discussion that we have dealt with, we now come to a kind of agreement that we need to know the thermodynamics of a protein molecule, the free energy changes that happens in your protein molecule, in order to decide whether it would be functional or it would be non-functional. Okay, we would also like to come on a consensus from the previous discussion that the protein would look for its lowest energy state so that it can acquire its functional activity. Okay, and from the Levinthal's paradox, we can now conclude that it is not necessary that the protein would follow all the possible path to achieve its native form or functional form. Okay, it kind of do not try out all the possible conformations. Some conformations can be skipped. Okay, so these are three most important concept of protein folding that you should digest before we begin discussing that ab initio protein folding problem. Okay, so now let us try to build up what is to be expected, or what is supposed to be done, or what is needed to be worked out for finding out the structure of a protein using ab initio approach, or what should be the main features of the algorithm that we are going to design that would predict the structure of a given protein using ab initio methods. Okay, let us now discuss that, right? So now if you want to follow the Levinthal paradox, we would require an efficient search strategy. Okay, so we would need an algorithm, or we would need a means, or we would need a method that would create a lot of candidate protein structures. Okay, so well, say for example, given sequence, we would need some computational method, or we would need an algorithm to have a feature that would create a lot of structure that this protein can adopt. Okay, what are the different possible structures that this protein can adopt? Okay, that would be the first main requirement for any ab initio protein folding algorithm. Okay, so we would call such predicted structures as decoys. What we would call them? We would call it as, this is decoy 1, this is decoy 2. We would call these representative structure that this sequence can adopt as decoys. So it is not necessary that these structures would be right. Okay, the decoys can be right or they can be wrong. All we want to do is to have an algorithm that would generate some set of structures that this particular sequence can adopt. Okay, so all in all, we want to have an effective search strategy. We would need a computational component in our algorithm that would search what might be the conformational state of a given protein B. Okay, so what can be the different conformational state that this protein can acquire. Okay, and the next next thing that your protein folding algorithm, ab initio protein structure folding algorithm, you might want to have is that there should be something in your algorithm that would delineate what are the good structures from the bad structures. Okay. There should be something in your protein folding algorithm that would help you discriminate what are the native like structures from the other decoys that have been predicted. Okay, so the scoring function would provide us with the confirmation that has got the lowest free energy state. As we know from the Anfinsen hypothesis, that the proteins are supposed to be at their lowest energy state if they want to be functionally active. Okay, so this would be the job of your scoring function that would enable us to determine that whatever candidate decoy structure that the search strategy has created, of which what are the lowest free energy states, what are the structures that are having the lowest free energy state, and thus they can act as good structures that can be used that can be considered to be functionally active. Okay, so these are the basic requirement for an ideal ab initio protein folding algorithm. First, we would require an effective search strategy, and then we would require something that would discriminate a good structure from the bad structures, or whatever, which are the good candidate structures, or which are the good, or you can say, candidate decoys that this search strategy has predicted. Okay, and that job is to be done by scoring function. So in our next lecture, we would discuss how are the search strategy implemented. Okay, and what are the things that are considered while defining the scoring function, how the scoring function was defined in order to delineate a functionally active form of a decoy uh, from the remaining decoys that has been produced. Okay, so this is all we would this is all I have for you people in this lecture. The next lecture we would start out with the molecular simulation methods that are usually implemented in search strategy as such, okay, or in ab initio protein folding algorithms. Okay, so see you guys in our next video.